Meiosis. In a eukaryotic cell, meiosis is an important process that takes place during sexual reproduction. In the beginning of the first meiotic division, the nucleus of the cell absorbs water from cytoplasm and starts to swell up. The volume increases threefold, and the cell is now ready to enter the first stage of meiosis, called Prophase I. During the first stage of Prophase I, called pre-leptotene, the chromosomes look like thin, uncoiled long threads. Later they start condensing and assume a definite shape. This stage is called leptotene. Unlike prophase of mitosis, two homologous chromosomes from each parent cell are attracted toward each other and they start forming pairs during the zygotene stage. In the pachytene stage, pairing of homologous chromosomes, known as synapsis, is completed. The bivalent or homologous chromosomes further undergo longitudinal contraction and become shorter and thicker. This marks the beginning of pachytene. Each bivalent chromosome is composed of 4 chromatids, therefore called tetrads. At this point, chromatids of homologous chromosomes cross over and they interchange chromatic segments, allowing more combinations of genetic material. The crossing over is accompanied by the chiasmata formation. The chiasmata are points where two non-sister chromatids exchange genetic material. The chiasma becomes very visible at this stage. This is the diplotene stage, in which the exchange of genetic material between the two homologous chromosomes occurs. Next comes the final stage of prophase in meiosis, called diakinesis, in which the chromosomes continue to pull apart and the non-sister chromatids remain loosely associated via chiasmata. The second stage in meiosis is called pro-metaphase I, which is marked by breakdown of the nuclear envelope. At this stage, the nuclear membrane disappears completely. Microtubules get arranged in the form of spindles between two centrioles and chromosomes get arranged on the equator of the spindle. During metaphase I, microtubules of the spindle are attached to the centromere of homologous chromosomes of each tetrad. The repulsive force between two homologous chromosomes increases greatly and chromosomes become ready to separate. Anaphase I. Chiasma separates and the chromosomes, each with 2 chromatids, move to separate poles. This stage is called Anaphase I. Telophase I. Nuclear membrane around the chromosomes is formed. This is telophase I. At the end of cytokinesis, 2 daughter cells are formed, after which the cell is ready to start meiosis II. During the first stage of meiosis II, also called prophase II, the centriole divides into 2 and each centriole slowly migrates to the opposite pole. Following this, the nuclear membrane disappears and chromosomes with 2 chromatids become short and thick. Subsequently, chromosomes get arranged on the equator of the spindle and the spindle attaches to them at centromere, marking the beginning of metaphase II. Then the centromere divides into 2 and each chromosome produces 2 chromatids. The microtubules of the spindle are seen attached to the centromere of chromosomes. During the next phase, called Anaphase II, by the contraction of microtubules, the chromatids start moving towards opposite poles. In Telophase II, chromatids completely migrate to the opposite poles. After this, the process of cytokinesis begins. It is the phase when the cleavage furrow begins and gradually deepens to form two daughter cells. This marks the end of meiosis II. To sum up the process of meiosis, we see that a single cell divides to form two haploid cells during the process of meiosis I. These two cells divide once more to form four haploid cells during meiosis II. Thus, during meiosis, a single cell gives rise to four haploid cells.